Good morning again. Thank you for coming back in for our first full session of the MoBE conference. Just one housekeeping note: there's apparently a bicycle chain in the front of the building outside. If it's anyone's in this room, please go remove it because the guards will remove it otherwise. I don't know if it was someone here or not, but just there you go.
I rode from Davis.
Oh, well, there's your bike's gone then. Jordan Peccia is going to start and chair our first session. And again, thank you all for coming. And online, thank you for watching. There you go.
Thanks very much, Lynn. So welcome to our first session entitled Microbiology of the Built Environment, Here and Now. I'm Jordan Peccia from Yale University, and I'll be the session's chair. So this session highlights the new research people and the new research programs and the depth of those programs from the MoBE program overall. Our first speaker will be Professor Jonathan Eisen, who's from the Department of Evolution and Ecology at the University of California, Davis.
I got my microbiology of the built environment bling here. So, all right, thanks. Thanks everyone for coming, and thanks to all the organizers and the Sloan Foundation and the National Academy. What I want to do here is talk a little bit about the microbiology of the built environment network briefly, and then talk about some perspectives that I've learned from this microbiology of the built environment program on the MoBE field. So I'm going to give a little bit of background on microBEnet, and then I'm going to talk about a few lessons learned that I think may be relevant to this program, and if you're thinking of starting a new scientific program, they might be relevant to that too, and then very briefly about future plans. So a little bit of background. MicroBEnet, the Microbiology of the Built Environment Network, was started in 2011 in response to a call from the Sloan Foundation. They put out a call to create this interdisciplinary network focused on communication largely. And we booted up the program and developed a mission for supporting, promoting, accelerating, and improving work at the interface of microbiology and the built environment. We designed our work to target three audiences, three sort of tiers. The Sloan MoBE program, that is people who were funded by and working in the projects funded by the Sloan Foundation in the Microbiology of the Built Environment program. Other microbiology of the built environment practitioners and people who worked in related fields like building science and microbial ecology and so on. And then what we generally called stakeholders. Broadly, we view this as sort of anyone else who might be interested in microbiology of the built environment, such as students and the public and funding agencies and Congress and so on. We developed a whole suite of programs that, again, largely focused on activities related to communication and collaboration. This included education, citizen science, organizing meetings and workshops, trying to help people track down information, deal with information overload. We developed a social media program, and we had a lot of focus on teaching people about the potential benefits of openness and sharing. I'm actually not going to talk an enormous amount about the microbiology of the built environment network activities specifically. If you're interested in more about what we do, you can go to microbe.net. We have lots of information there about our current activities, social media activities, meetings and conferences, resources that we've developed or other people have developed, and so on, and you can go there. I want to talk very briefly about one thing we've been doing recently which relates to this meeting, which is with the help of the Sloan Foundation and Julia Maritz and David Coyle at microBEnet, compiled a list of publications supported by the Sloan Foundation funding, 315 papers in total. Of those papers, there are 1,633 authors, and then we got help from the UC Davis library team, Daniel Hicks and Carl Stammer in particular, to do some preliminary bibliometric analysis of this, and we're continuing to work on this. Just to give you a little insight, this is a graph of the publications by year, and as Paula already talked about, there were some publications early on, or publications supported early on that showed up later, but there's obviously been an increase in publications over time. This is a word cloud of the journals where the papers have been published. So there's distribution across different journals. PLOS One had the most papers. Indoor Air, I think, had the second most papers. Microbiome I think had the third most papers. There's been a distribution across a reasonable number of journals with some weighting towards a couple of them. We've been looking at citations to try and assess one metric for assessing impact of these publications. In total, there have been so far 16,316 citations. In our original analysis, a little higher if we add in one or two more papers that didn't show up in the original analysis, but something like that. An H index of 49, I-10 index, that is the number of papers cited more than 10 times, of 136. As you would expect, the number of citations is related to the year of publication. So someone asked this question before about assessing impact and how you can assess impact of sort of recent developments. It's easier to assess impact of older things than newer things. There are a few papers that have more than 100 citations. I think it's 13 or so papers that have more than 100 citations. Many of these are focused on methods development. That's common in the scientific literature. We've done a little bit of analysis of, we're really interested in pursuing this further, and I just want to give you a little taste of this, of co-author networks. And that's why I needed my computer here, if I can switch over. And I will post this HTML file for people to play around with. We have a co-author network of all the key authors, PIs funded by the community, and who else they are publishing with, both within and outside the community. If you zoom out, most of the people are connected to each other in some way. Some people are connected more than others. And again, this is really preliminary analysis. We're going to be pursuing this, and if people are interested in this type of work, please contact me. We're posting all the data, and we'd love to get participation from other people in this. So I just want to briefly talk about some lessons that we've learned from this, that I've learned from this project. The first lesson that I've learned is that people are everything. The people that are involved in these projects are critical to their success. At UC Davis, we've had a large number of people over the years involved in the microbiology of the built environment program. This includes senior personnel, PhD students, multiple collaborators within UC Davis, and an army of undergraduates, largely supervised by David Coyle, who's back there. Is it really your birthday today? It's David's birthday today. David's done an amazing job of wrangling the undergraduates in the lab and getting them to do things related to microbiology of the built environment. Here's a list of the army of some of them. We've had lots of external collaborators, in particular Hal Levin and Shelley Miller, who've been directly involved in some of the microBEnet activities, as well as many other collaborators who've been involved in some individual projects at microBEnet. And if you go to the blog, you will see that we've had hundreds of guest bloggers at microBEnet. If you're interested in becoming 101, 102, et cetera, please let me know. Ed Yong talked yesterday about the stories of microbiology. We really believe in the importance of stories, and we want people to tell the story behind their research. So if you publish a new paper and you want to share the story behind the research, please contact me. It's a great way to get more eyes on your paper and get people to see the person behind the science. Another thing that's really important is the importance of history. As an evolutionary biologist, I'm kind of obsessed with history. That's not the part of history I'm talking about here. And I'm going to do some self-humiliation in the next minute. So I was quoted in a newspaper story. It actually was an accurate quote of something that I wish I hadn't said. We know nothing about the ecology of microbes in buildings, says Jonathan Eisen, principal investigator. And Noah Fear, who I don't think is here, but Noah Fear posted on Twitter, like, what the... That's not true. We've been studying microbial ecology of buildings for more than 75 years. And that's in fact true. I even have a collection of books on this topic, including this one, Bacteria in Relation to the Country Life. It's really important when you're building a new field to not portray it as completely new, if there is context. There is a lot of rich context of microbiology of the built environment. Maybe they weren't using the same methods that we're using now. Maybe they didn't have the same volumes of data that we have now. But people have been studying the microbiology of the built environment for a long time. And it's important to recognize that history, cite it, and appreciate it. Another thing I've learned is that diversity matters. There are many different aspects to this. So microbial diversity, the taxa, the genomes, the functions that are found in microbes, most of them are pretty poorly characterized across the planet, including in the built environment. We've done a lot more work in most environments on bacteria, less work on archaea, fungi, protists, and viruses. There's still an enormous amount we don't know about the diversity of taxa, genomes, functions present in the built environment. Obviously, the diversity of built environments is also still in its early stages of being characterized in detail, the type, the location, the materials, etc. A diversity of approaches that people use to study the microbiology of the built environment. And one thing that's really, really important is the human diversity, the diversity of the occupants, the diversity of the researchers, and the diversity of the participants. And we should not neglect that, and we should try and emphasize broad diversity. If we want to build a field, being inclusive in that field is really critically important. Another thing that, as a communications project, not surprising that we've learned that communication matters, or that we think that communication matters. We believe fundamentally that social media can be a powerful tool in scientific and engineering communications. It can be used well or poorly. It's like a Xerox machine. You can take pictures of weird things or copy stuff that's really important. It's important to learn how to use social media, but it can be used in many powerful ways. And again, if you're interested in social media and the built environment, please contact me. We'd be happy to help people develop programs in it. Platforms for community building are still limited. So we've been developing a lot of sort of web tools and platforms for putting together the community here. There's still not a lot out there for doing this robustly in other environments. Relating to Ed Yong's talk last night, work with the press, really important to communicate not just with the sort of your colleagues and the public, but also with the press. Don't oversell what you're doing, but it's really important to do that communication. One thing that we've learned that's really critical is that the language and culture in different communities is really important to deal with from the beginning. Architects, building scientists, engineers, microbiologists, bioinformaticians talk very differently, act very differently. It's really hard to get them all on the same page to communicate about things. And it's really important to tackle that from the beginning. Openness, open scholarship, open access publishing, open data, open source software, really critical towards embracing and bringing in other people into the field. One really important part of microBEnet activities has been to try and survey what's going on in the microbiology of the built environment community and identify gaps that exist. So Paula already said that the Sloan Foundation sort of views one of their tasks as to identify gaps in particular areas and support them. We were now identifying gaps within the microbiology of the built environment field, and we ran a series of workshops on some of these gaps, and I think it's really important to keep thinking about this in any field, get people together to talk about those gaps and try and do something about it. We've identified many areas that we had workshops that led to, I think, very important and successful results on citizen science, on living or dead microbe identification, on animals in the built environment, on reference genomes and the need for reference genomes in the built environment, on studying space vehicles and related activities, on developing standards and methods, and there's still lots of gaps that exist that are out there, and there's still room for organizing people in this community, so I think people should think about that and try and continue to talk to each other about what the gaps are and organizing the community to try and fill in those gaps. Education and engagement is critical to the building of a new field, to bringing in new people in the field, to bringing in the public. Public engagement, for example, via citizen science has been particularly successful, I think, in microbiology of the built environment and related fields, and needs to be expanded and continued. Research-based courses are really important. We've been developing at UC Davis this Swabs to Genomes program where we have students come in, swab some environment, isolate and identify organisms, and then eventually sequence the genomes of those organisms in the ideal world, and even publish papers on those. We've published a protocol for doing this. Every step of the way, everything you need to do this process is published, but there aren't a lot of sort of general course materials out there for studies of microbiology of the built environment, so I think it would be useful to develop more of those. What I call edutainment is okay and even good, and I forgot to bring up, David, did you bring any extra copies of the game today? We developed a board game called Gut Check on the microbiome that actually was produced and released by MoBio, which has now been bought by Qiagen, but if you're interested in playing about microbiomes, doing fecal transplants on yourself or your neighbor without the grossness of fecal transplants, let us know. Again, courses and course materials would be really helpful to develop and release and share more of those for microbiology of the built environment. And lastly, I just want to talk briefly about what our sort of future plans are. So we've been thinking about microbiology of the built environment and microBEnet as the Sloan Foundation is wrapping up funding for this community. We have a couple more years of funding from the Sloan Foundation, and what we're doing in part in these last 2 years is trying to streamline our activities and think about how we can continue to get support in the long run for our activities, even though the Sloan Foundation won't be funding that work. So as part of this, sort of our general principles for doing this are to sort of maintain the core activities, probably reduce many of the peripheral things we've been doing because some of them are pretty expensive, but maintain the core activities. But keep broad targeting of activities. And in particular, what we want to do is expand, in essence, beyond microbiology of the built environment to include all sorts of other themes which overlap with microbiology of the built environment, but to include much larger audiences. So for example, antimicrobials and antibiotic resistance, a really big topic in the built environment, an even bigger topic outside of the built environment. And so we're trying to develop a whole program in running communications about microbiology of the built environment and antimicrobials and antibiotics, but also making it broader than microbiology of the built environment. And we have a collection of these themes that we're working on that are on the website that we're trying to develop into new potential areas. This includes things like space microbiology, water systems, again, not just the microbiology of the built environment of these, but expanding beyond those core areas. And so I'm really appreciative of all of the people are critically important in any program. In microBEnet, the people have been everything for microBEnet. We're not doing a lot of research. We've had small sort of little individual research projects. Most of what we've been doing have been about communication. And this communication relies on a lot of people to do that communicating or to work with the community. David Coyle has been sort of the main instigator of a lot of the microBEnet activities. We've had a lot of other people involved. And again, the internal and external people have been critical to everything that microBEnet has been doing. And I also want to acknowledge the Sloan Foundation and Paul Olszewski, and I spelled your name wrong, that's awesome. Typing at the last minute, that's brilliant. I think she's been really instrumental in getting us to think about not just what we can do for sort of communication activities, but how we can think about the long term of our program, assessment of our program, and whether or not what we're doing is useful beyond just the core collection of MoBE researchers to the broader community of scientists and to the country and the world. Thanks.
So we have time for just one question.
Yell, Shelley.
I want to know if would you do this again all over?
Yeah, so would I do it again? Absolutely. I mean, when Paula sort of sent me the announcement from the Sloan Foundation and said we're trying to recruit someone to apply for this network activity, I was a little not convinced because I've done all this on the side previously, but I've never actually had an official project on sort of communication and networking, and I was initially reluctant, but I would absolutely do it again. It's revolutionized what we do in my lab in hundreds of ways, and that's why we want to keep going with microBEnet activities, both in the built environment and in related areas to the built environment in the long run. I would absolutely want to continue this, and I would drop many of the other things we're doing to keep doing this type of thing.
So please join me in thanking Professor Eisen for this fantastic MoBE resource, and also I think of really particular interest is the quantitative tools that he showed early on in his talk that are going to track progress of MoBE program into the future. Thanks, John.